Okay, so I'm assuming you guys can hear me. If you guys can hear me, please say something in the chat. If not, let me see. Can you guys hear me? Okay, perfect. تمام. Okay, so راح احاول ما اطول عليكم كتير. انا بعرف انه هذا الموضوع جديد عليكم. بعرف انه اكيد نزلتوا البي دي اف وطلعتوا بالسلايدز وقلتوا يا ربي شو القصة وما عم نفهم شيء وما بعرف شو مكتوب بالسلايدز وكيف بدنا ندرس. So don't worry about that. يعني ماكسيموم بعطيكم ترانسكريبت من اللي عم نحكيه. بس انه المفروض كل شيء يكون مشروح هلا باللكتشر. So don't worry about that. خليني اشوف الصوت واطي. Is it okay now? هلا الصوت مليح? I think now it should be good. Okay, perfect. So, DNA sequencing. I think, yes, it will be recorded. YouTube Studio automatically records everything and it just uploads it. بس نخلص I will give you the link ترجعوا تحضروا. Okay, so let's go to the actual topic. Okay, so why do we want to sequence DNA? وليه عم بحكيكم قبل ما نحكي ليه بدنا نعمل sequence للدي ان اي؟ بدي احكيكم ليه بيهمكم انتو as biomedical scientists انه تفهموا عن DNA sequencing. So there are two things. First of all, انتو منكم طلاب مختبر. Into biomedical scientists, so you need to know about the biomedical sciences. مش بس you're not the typical employee بروح على المختبر بكبس 3 زرار بيطبع ورقة بيدخل فحوصات لمريض. Okay, you're way more important than this. لو بدنا اياكم تكونوا طلاب مختبر في تروحوا على المهنية بتدرسوا شوي صغيرة كيف تكبسوا لكم 3-4 كبسات على كل مكنة. And then you don't need to interpret anything. No, you guys are way more important than this. هذه أول شغلة. شغلة تاني هي متوقع إنه من 5 سنين لقدام ل10 سنين, 5 to 10 years from now, معظم المختبرات الكبيرة إذا مش كل المختبرات الكبيرة رح يكون عندهم somehow to sequence DNA. And this will bring me to the question of why do we need to sequence DNA? لأنه we are different. نحن أشخاص كتير مختلفين عن بعض. And DNA sequencing allows me to tell exactly what will work for you وأي شيء ما بيشتغل لإلك. The best example I can give you هذه بشوفها تقريبا يوميا my practice as a medical doctor. هو there's a gene اسمه MTHFR. It has to do with folic acid and whether you can إذا فيكم تاخدوا folic acid and you can metabolize it or not. And a mutation in this gene can put you at a risk of many things. ما رح فوت بتفاصيلها هلا. It's not part of the course, but I do want you to understand إنه once I know your sequence of DNA, I know exactly أي نوع folic acid رح أعطيكم إياه. Okay, so رح أحكيكم شوي عن the history and the current practice, and this is what you need to know. ومننهيها آخر شيء a conclusion. رح نحكي فيها إنه the potential for use and the potential for abuse. Okay, so what is DNA sequencing exactly? DNA sequencing معناتها إنه هو it's a general laboratory technique. بدنا بالضبط السيكونس of nucleotides. أو النوكليير بيسز يلي موجودين بالدي ان اي موليكيول وهول اللي عادة بنسميهم ATCG يلي فيها كل البيولوجيكال انفورميشن يلي الخلية بتستعملها حتى تشتغل، حتى تعمل development، حتى تنقسم. and eventually لما يصير عندنا معلومات كافية عن ال DNA sequences we can create something known as a genome. لما نحكي عن الجينوم مقصود فيه انه اكترية ال DNA يلي موجودة بجسمنا we now can read it. رح نحكي عنها بعد شوي كمان. and وهيدي شوي بلشت تتطور. يعني مش دغري وصلنا للي نحن وصلنا له. It took a lot of time of research and development to get to where we are today. So I'm going to give you an example of importance. تقلكم لأي درجة مهم إنه to sequence DNA. Okay. Now, once you sequence DNA, you can translate it into a protein, and that's why you'll find these are very short. These are protein sequences, but the original was DNA. We just changed it into a protein sequence حتى نوفر مساحة. Okay. Now, this is a file. هذا الفايل basically أخذ a virus sequence أيام الكورونا من مدن مختلفة حول العالم. So, for example, we have the sequence of الفيروس يلي ضرب ببيروت، الفيروس يلي ضرب بشنغهاي، بالقدس، بطهران، etc., right? And so we have the ability by using computer magic, by using something known as bioinformatics, to be able to sort of see المutations يلي عم تصير كل مرة. And eventually we do our computer magic and you get something like this. You get something known as a phylogeny tree. The phylogeny tree allows you to tell انه اوكي على راس التري من فوق في عندكم جيروسالم يلي هي القدس اورشليم. من بعدها في عندكم التري لوحدها في بقلبها ليما بيرو، واشنطن دي سي بالولايات المتحدة، لندن بالمملكة المتحدة. بعدين عندكم باريس وبيروت تقريبا ذات السترين. This makes a lot of sense لأنه في كتير أشخاص بيجوا مش بس من فرنسا، بيجوا من أفريقيا عبر فرنسا. So بهذيك الايام هيك كانت. فطبيعي جدا انه الاشخاص اللي قطعوا بباريس هن اللي بعدين يعدونا ببيروت. And then Cairo and Damascus، وبالاخر عندكم طهران is alone. So الاصل كاين من جيروسالم. And this is how we knew من وين اجى الفيروس لعنا eventually. Right, and we know إنه إذا علاج معين بيشتغل بأمريكا مش ضروري يشتغل عنا، بس إذا اشتغل بفرنسا على الأرجح بيشتغل عنا, etc. So this is the importance of sequencing DNA. This is one of the importances. أكيد في له أهمية أكتر بكتير من بس هذا الموضوع. Okay, so the other reasons to develop DNA sequences لأنه you can detect diseases early. And I'm talking about genetic diseases يلي ممكن يبينوا بعدين. مثلا عم نحكي عن Alzheimer's، عم نحكي عن Huntington's disease، عم نحكي عن diabetic predisposition، إمكانية إنه واحد يصير معه أمراض وراثية معينة، أمراض مزمنة معينة. نحن بنعرف إنه there are some DNA sequences some DNA sequences that are associated with certain diseases. And these diseases للأسف ما فينا نلقطها بالconventional methods. لما نقول conventional methods أنا قصدي الأشياء اللي أخذتوها بالهيماتولوجي، بالبارازيتولوجي، clinical chemistry, things like that. You cannot do a test to guess إذا شخص يحصل معه Alzheimer's. You need DNA sequencing, right? And finally, there is something known as individualized medicine يلي هي كل شخص لأنه مختلف عن التاني. It makes sense إنه نعمل a protocol مختلف عن التاني. And I'll give you a simple example. هلأ شخصين يكونوا عندهم وجع راس. هو الشخصين كل واحد نعطيه حبتين بانادول، باراسيتامول 500 ملغ. Okay, so it's 1 gram of paracetamol كل واحد أخذ. واحد بيوقف وجع الراس، وواحد تاني ما بيروح وجع الراس. Okay. So we need individualized medicine, and if you know the DNA sequence, this helps you a lot to do individualized medicine. So let me give you a bit of history before we start with the actual DNA sequencing. بال 1980 Uwait Waikan et al. They suggested a prenatal genetic test للثلاسيميا. واعتبروا وقتها انه بدنا نعمل genetic test بس ما يكون معتمد على DNA sequencing لانه بعدنا بال 1980 and so it's not really very feasible to do genetic testing and they sort of succeeded and the whole idea was to test the fetus. ناخد شوي من الامنيوتيك fluid اللي بسموها مية الراس. We test it and then we see if they have thalassemia or not. And this was supposed to be, depending on which allele they have, we can tell if they are thalassemic or not. Except that it was too expensive, it was too dangerous, and they did publish the paper in 1980 لأنه ما كان في بديل. بس تخيلوا اليوم عم نحاول نعمل ذات الشيء. أكيد في طرق, that are much better. And so this is why we have to talk about DNA sequencing. Now, before we talk about DNA sequencing, this is very important. I know for sure Dr. Alim Tayrik رح يحكيكم عن ال PCR بتفاصيلها, the very intimate details. But for you to understand DNA sequencing, I have to explain a bit about PCR, our polymerase chain reaction. And once you understand it, you'll understand how we sequence DNA, how we can do this. Okay? So, very simply, تفهموا ال PCR أو ال polymerase chain reaction, which has been like ما في حدا ما بيعرف شيء ال PCR بهالأيام لأنه صارت من وراء ال COVID-19 أو ال SARS-CoV-2 virus انتشر بين كل العالم. But what you need to know is that PCR amplifies a very specific region of DNA strand, right? بنسميها the DNA target, and you need to know what the target is, or at least you need to know part of the target يلي بنحطها بالprimers. And so for PCR to work, what you need is أول شيء you need a DNA template, right? A piece of DNA, and you need a polymerase, which يلي هو an enzyme يلي polymerizes new DNA strands. This polymerase should be heat resistant. That is, we use the TAC polymerase, TAQ, Thermophilus aqueous. And we need 2 DNA primers. يكونوا complementary the 3' ends of each DNA target. And of course, you need buffers, you need the triphosphatates, you need the DNTPs يعني. And you need magnesium for this to work. And then you just run the PCR. You keep it's a cycle of نفصل ال DNA عن بعضها, بنركب البرايمرز, نعمل extension, and then we repeat. نفصلهم عن بعض, نعملوا extension, and you keep repeating over and over and over, right? And you need very, very small volumes for this. تقريبا you don't need a lot of volume. 10 to 200 microliters in small reaction tubes, you'll be fine. So between PCR and DNA sequencing, مرقنا بفترة سميناها بفترة كنا نعمل شيء اسمه comparative genomic hybridization, which was very, very easy. You take a genome that you know is normal, or at least you're hoping is normal, right? And what you do is you paint this genome green, and you take the genome that you want to sort of study, and you paint it red, and then you just mix them together, and then you look at them under a fluorescent machine. تمافت بتفاصيلها. And so what you do is, what you're doing is when you paint it yellow, you use DNA sequences, known DNA sequences that have a fluorochrome, that they are tagged. وبهالطريقة بيكون مثلا التاج لونه أحمر أو التاج لونه أخضر. And so what we do بهذه الحالة هي إنه we use a fluorescence microscope, special computer programs to do this, and then you look. إذا بتشوفوا بالصورة في عنا هون منطقة 2 3 حمر ومنطقة خضراء. It means المنطقة الخضراء معناتها only the normal genome عطى a signal. معناتها something was lost from the patient يلي كان المفروض يكون أحمر. نطلع بالمنطقة الحمراء معناتها we have extra genetic information, and so we have like trisomies perhaps. هون it's a trisomy لأنه it's multiple dots, but you'll also find little dots بين فوق الخط الأصفر. These are like okay, this has been duplicated in the patient, and then of course if you mix red and green light, you get yellow light, and that's why yellow means normal. This was good. This was not a bad idea. من ناحية إنه I know a bunch of diseases, I know their sequences, and let me test for them. But it's not enough. Like you can test for trisomies. فينا نعمل test إذا في حدا عنده مثلا an extra copy of the gene, or a deletion, for example. إذا بينت نقطة خضراء بدل ما يبين بالمنطقة الصفراء. But there was a better way of doing this, like a much more efficient way of doing this, and the much more efficient way of doing this was by using something known as microarrays. So بدل ما نعمل اللي حكيته هلا يلي هو انه نطلع على الميتافيز ونقعد نفتش على الكروموزومز وهيك يطلع الكروموزوم مقطوش او for whatever reason, let's bind to a glass slide. You bind these specific sequences that we choose. معظم المايكروتشيب arrays they can bind 9096 sequence, different sequence أكيد. And so what you do is you get a person's DNA and the reference DNA, and you put them both on the microchip itself, and then eventually you get something like this. You allow hybridization. إذا كان لأنه what you really have is depending on the locus you get a different result, so one color for each allele. And if you have two greens معناتها you have two correct alleles. If you have two reds معناتها you have two incorrect alleles. And if you have a red and a green معناتها you are heterozygous, you have a heterozygous mutation. And that really worked for a while. وكان ماشي حالنا فيهم. The main problem was that you had to wait until you had enough patients لأنه one microchip can do sometimes up to 40 patients, sometimes even more حسب التكنولوجي, and they require a special scanner مثل اللي شايفينها هون. This is from Illumina. It's a very specific scanner, and this is how the results usually look like. So you have a picture of the microarray, which is in the background, and then you will find that it gives you a full list of what is expected and what was the result, which is really nice. The computer does the work for you, ومش مضطرين إنه تقروا into the actual result. So. But that was nice, right? You can do this, but it's not enough. And the reason it is not enough is that there are DNA sequences نحن ما بنعرفهم, they are linked to certain diseases, and we need to sort of discover these things. And so when DNA was first sequenced in 1977 using شيء اسمه the Sanger method, لأنه a scientist whose family name is Sanger اخترعها, it used to cost $100 million per genome. So if you want to do your genome, you get a check for $100 million وبتدفعوه للشركة وبيقعدوا شي عدة سنين. It took them quite a while to do a genome. Something like 3 or 4 years, and 3 or 4 years later they tell you, hey, this is the sequence of your DNA. have fun reading it. DNA sequences are like من ناحية المعلومات they're not very very long. من ناحية as far as computer information is concerned, like you can download the entire human genome project اللي رح نحكي عنه هلا. بيطلع كله على بعضه شي 10 gigabytes, which is nothing compared to other things. Okay, so in 1990, they decided we have to do the entire genome of the humans. And already you can think of many problems with this. You need at least 2 humans. You need a male and a female. هذا أقل شيء ممكن نعمله. هذا بعد ما حكينا بالvarieties of humans. So شخص من الشرق الأوسط غير من شخص من أفريقيا غير شخص من أوروبا غير شخص من اليابان غير شخص من الهند. And so بدنا عدد كبير من الأشخاص حتى نقارن whole human genomes. And it took them 11 years to sort of complete the human genome project. The human genome project wasn't completed until very recently. بعدين نحكي it was completed actually in 2006. بس نحكي شوي عنه بعدين. So it cost them تقريبا $100,000 per genome. The entire genome project كلف $3,000 مليون دولار, which is 3 مليار دولار. And so it cost them around $100,000 per genome. By 2005 صار عندنا شيء اسمه second generation sequencing رح نحكي عنه, which will cost around $10,000 per genome. And then eventually what we have today ببعض المختبرات موجودة بلبنان بمختبر اسمه إينوفا موجود ببعبدة, which is third generation sequencing, which will cost you somewhere between around $1,000 per genome. ممكن يكون أرخص, depends on the technology that they are using. Okay, so the Sanger method. The Sanger method is the reason ليه قلت لكم إنه لازم إنه basically لازم تعرفوا شوي عن ال PCR. لأنه essentially if you take PCR ومن زعبل شوي صغيرة you get essentially the Sanger method. Okay, so what does that mean? You know how in PCR you have your primers, and then you have the polymerase بيركب على البرايمر وبيكفي ساعتها. YouTube is going to show you an advertisement. This is something ما بقدر أهرب منه, unfortunately. So after the ad برجع بكفي لكم نص سانجر. Okay, so the ad has been inserted. I'm not sure is بيان لكم or not. Anyway, I'm going to continue. Okay, so here's the thing about Sanger. What if we had a special dNTP? This dNTP, what's special about it is that the moment بيركب مع DNA sequence, the synthesis stops. The polymerase will stop. Okay? When بيركب, it's sort of like random. شوي عشوائي. يعني ممكن يركب بالأول, ممكن يركب بالآخر, ممكن يركب بالنص. It doesn't matter. لأنه ما تنسوا عم نعملها هاي العملية ملايين المرات, أو آلاف المرات, مش ملايين المرات. Okay? And so let's put, خلينا ناخد 4 tubes. We do كأنه PCR بالأربع tubes, except in tube number 1 بدي أحط DDATP, بتيوب number 2 بدي أحط DDCTP, tube number 3 has extra DDGTP, وتيوب number 4 في عندكم extra DDTTP, right? So only one of the DDNTPs is put in each of the tubes. And so polymerase راح يبلش يعمل sequences that always end with, so let's take tube number 1. Tube number 1 في بقلبه sequences that always end with an A. And these sequences have different sizes لأنه it's sort of random, right? عشوائيا قاعد ال A. Now, for the primer, we know exactly beginning sequence when we want to DNA sequence. The reason we know it is that we use restriction enzymes. The restriction enzymes, they splice at a very specific sequence. So بنعرف وين انقطع وبنعرف شو البداية. So مش عاطلين هم نحن البداية. بنحط برايمر من الجهتين وبتبلش يعمل DNA sequencing, and eventually if you run them on a gel you will get something like this. Okay, now remember the first column is A, then T, then G, then C. هذا الترتيب اللي لهم. So if you look at them نقراها من فوق لتحت لأنه فوق هي أصغر قطعة وتحت هي أكبر قطعة اللي ما قدرت تمشي كتير. So if you look at this the example that I'm giving you نحن قلنا إنه الترتيب اللي لهم is going to be ATGC. So the first letter from the top بين مقطوش شوي which is T. من بعدها you will get a C and then a G and then T, T, A, T, etc., etc. So now we can sequence them, right? Because you have these short fragments that were terminated sort of randomly. And then you can go back reading the ladder وبيطلع معكم the sequence. Of course, this it's a beautiful method. It's very nice. It uses radioactivity, which is not so nice. بس المشكلة فيها okay, so just to answer a question, yes. So to give you an example, the ddNTP is sort of like cut it randomly, but at that letter itself. So let's take tube number 1, يلي كان في بقلبه الA's, and I'm going to show this in the chat. Okay, let's say our this is our sequence, for example. Okay, and so this is our sequence: ATA, CG, ATC, GA, for example. And so you will have fragments that look like this, ATA, and you will have fragments that look like this, ATA, CGA. And then you will have fragments that are complete, لأنه it ends with an A, so you will have these fragments, right? And so ترتيبهم بيطلع ترتيب مختلف على الجل يلي عم نشتغل عليه. Okay, and because each one of those ddNTPs has a radioactive isotope, we can actually see it on the gel. And this is how Sanger did it the first time. وين المشكلة بهذا المشكلة بهذا التكنيك إنه أولاً the first 15 to 40 bases they are of poor quality. شو يعني of poor quality؟ بتعرفوا لما تسكروا السحاب والسحاب ما يسكر على الآخر يبقى مفرشخ شوي من فوق ومن تحت. This is exactly what happens with DNA. And so you sort of get this problem at the beginning. صحيح بيلزق مظبوط البرايمر بس بعدين بيرجع بيفرشخ and it doesn't work. The second problem is that you can only do very short reads. And when I mean short reads, يعني the typical read is around 600 bases. Anything beyond 700 to 900 bases عم نخاطر. Anything أكتر من 900 bases, good luck. There is no way you can read this using the Sanger method. لأنه الجل بحد ذاته ما بيقعد بيساعن. And this was a big problem. ما تنسوا الهيومن جينوم بروجكت that was created عملكم بين 1990 لل 2006 most of the times was using the Sanger method وعذاب. That's why it took them so much time to do it. لأنه they have to splice the hundreds of thousands. لأنه remember إنه our genetic material is about 3.2 billion nucleotides. So they had to break it down into 600 nucleotide fragments and then do it over and over and over again, right? It's not something easy. So the Human Genome Project it's a collaboration ل 20 مؤسسة من research institutions. It's spread across 5 countries. كلفهم $3 billion. And in May 2006, they published in Nature magazine the sequence of the very last chromosome, and they considered it sort of complete. Now here's the thing. When I say it's complete, it's not exactly complete. Okay, the Human Genome Project only sequenced the euchromatin. It did not sequence the heterochromatin, right? So the Human Genome Project اللي هلا فيكم تفوتوا عليه من ال NCBI database. It's available for free online. It contains تقريبا 90% of the 3.2 billion nucleotides. It contains 2.9 billion nucleotides, and the rest is there's 9% that was sequenced بعدين, and the 1% is impossible to sequence لأنه there are so many repeats, and once you have a lot of repeats, it's impossible to almost sequence them. But it doesn't matter لأنه we don't care about them. نحن بيهمنا الeuchromatin, not the heterochromatin. And so, but it's to give you an idea, it's almost impossible to sequence very repetitive DNA using current techniques. So what was it used for, or what is it used for? This is a very interesting thing that happened in 2009. The doctors and researchers بالchildren's hospital بالmedical college of Wisconsin كانوا أول فريق بالعالم to sequence all of a person's genes, and they used the information to pinpoint a new disease. It took them about 4 months to do the lab work, and eventually they were able to save the life of the child. And so they were hoping, this technology will help save people from coronavirus, which was, to be honest, very, very interesting. Now, من بعد السانجر method, and this is something, you should know the Sanger method by now. You should know also the second generation sequencing. So I wanted to imagine Sanger method مشكلته and it requires 4 tubes and each one runs on a lane. But what if instead of putting radioactive isotopes بنحط fluorochromes, so something that lights up a different color. Let's color A, for example, red, T ملونة yellow, different colors, different fluorochromes, and put them all in the same tube. And then we just run the result. You do the whole PCR thing, you do replication, DDNTPs, they will stop مثل بالسانجر method. بس الفرق هو إنه بدل ما نقعد نعملها على electrophoresis gel, نستعمل شيء اسمه capillary electrophoresis. And it works. It works really well. And so what you will get is this, okay? This is how the result will look like. على جهة الشمال there's the raw data which doesn't make any sense at all. على جهة اليمين هذا أنا عم بشتغل على الجل electrophoresis machine. We were sequencing a genome like a patient in Spain, and in the middle you get the beautiful, the glorious DNA sequence, and this is given as something known as a FASTA file or a FASTQ file, depending on the machine. It not only tells you the DNA sequence, but it also tells you the quality of the DNA sequence. Okay, it's a text file في بقلبه ال DNA sequence, تحته في sequence of letters that tell you whether the DNA sequence was good or not, وقد ايه فينا نوثق فيها. Okay, so which brings me, أحكيكم باختصار عن ال second generation sequencing, لأنه we don't use it anymore these days. الصورة اللي فرجتكم إياها هي من 2016, but recently حتى بال 2016 كان عندنا الإيلومينا بالمختبر بإسبانيا. we used it all the time. بلبنان قلت لكم موجود الإيلومينا بمختبر اسمه إينوفا. ببعبدة, and they use it all the time. موجود بالAUBMC in their labs. And I expect the next generation sequencing يكون موجود تقريبا بكل المختبرات, because these will become cheaper and cheaper, and they will become more accessible to researchers and the patients and the doctors that will use them. So من هلا من 5 سنين ل 10 سنين من هلا متوقع إنه تكون عم تشتغلوا some sort of next generation sequencing. So what is next generation sequencing? This is the latest technology with DNA sequencing. There is a third generation sequencing. They're developing it. Error rates are up to 40%. That's why we don't use it yet. بعد قيد التجربة. بس حاليا أجدد شيء the top of the line technology is next generation sequencing. It doesn't cost a lot. ممكن تكلف بين $2,000 ل $5,000 per genome, and the reason there's a variation, it's because it depends what genome are you doing. So a human genome بيكلف بحدود $1,000. إذا عم تعملوا genome ل a virus, it's going to cost you closer to $200. إذا عم تعملوا genome ل a plant, okay, plants have sometimes 40 pairs of chromosomes, في منهم they have 50 pairs of chromosomes, you're going to push up to $5,000. That's why it's a very wide range. Depends شو عم تعملوا. مثل كل ال DNA sequencing, it is based on PCR. It is a modified PCR with wizardry and هيك خزعبلات صغيرة هلا بحكيكم عنها. I'll show you exactly how it works. And it uses a technology اسمها massive parallel sequencing. And massive parallel sequencing معناتها we're not just sequencing one strand, we duplicated the strand so many times and we sequence all those strands together and we get a consensus. الأكترية شو بيقولوا وهذا بيطلع اللتر المضبوط, لأن there's always a chance for mutations, there's always a chance for problems with that. And there are 5 methods from 5 different companies. ما كتير بيفرقوا بين بعض. They only differ in like some technology. التكنولوجي بتفرق من ناحية الباتنتينج. So واحد منهم مثلا الإيلومينا uses colors تيعرفوا أي حرف. Ion Torrent, which is another corporation, بيستعملوا electric pulse تيعرفوا which letter they are reading. But they all have almost the same concept when it comes to replication and when it comes to reading the sequence itself. And so the 5 available methods هن شيء اسمه sequence by synthesis, there's pyrosequencing, there's ligation mediated, there's phospholinked fluorescent nucleotides. I'm not going to go into the details, but I want you to know that it's like ما في one method. There are 5 different methods. اوكي رح احكيكم عنها. هي تاعت شركه إيلومينا يلي هي اكثر شيء موجوده حول العالم. And this is the one that هذه الموجوده بلبنان كمان, and this is the one اللي عم تنتشر كثير حول العالم, and this is the sort of easiest to understand من بيناتهم. Okay, so very simply, break down DNA. بعدنا كأنه عم نعمل Sanger method, by the way. It's the same idea. You have to break down the DNA somehow. We use restriction enzymes, and we know ما بيهم الterminal sequences لأنه this is what we want to discover. But what we do is we attach 2 adapters into each sequence. This is the important part. هون بلشنا نختلف شوي عن السانجر. So we attach a sequence of DNA that we know its sequence لأنه we synthesized it, and it works like a barcode. It's unique per patient, unique per run, and sort of like we have to know its sequence. And so these adapters, they have a region عليهم غير إنه they have to be unique. There's a region on them that allows them to stick on adapters. So if you get the slide يلي رح نعمل عليها الإكسبيرمنت, في عندكم little adapters مثل شعيرات صغيرة طالعين من الglass. There's a technology, there's a way to do this. It's very technologically advanced, but they polarize the molecule and they allow it to stick on the glass itself. And so what you get is you allow the adapters to stick on the probes on the slides. And once they stick, the polymerase تعمل a new strand من اللي لازقة ب the probe itself, and the double-stranded molecule is denatured and it is washed away. Okay? لأنه I don't care about the molecule, I want الادابتر إنه يكفي السيكونس اللي له. So right now كأنه اللي عملته من بعد الريبليكيشن is لزقنا كل هول ال DNA molecules اللي قصقصناهم onto the glass. And the glass and we force the DNA إنه to fold over وتركب على other adapter. And then we give it polymerase again. And then it replicates. And now you have all the strands, all the glass probes صاروا into complete strands. And you repeat it over and over and over and over. اللي صار عندكم شيء بنسميه clonal amplification. You amplified your DNA by creating clones. And now we start the reading itself, okay, which is this one, which is known as sequencing by synthesis. The polymerase will begin synthesis of the DNA باستعمال special DNTPs. These are not مثل Sanger DNTPs that stop the reaction. No, these are special DNTPs. Once one of those special DNTPs بيعلى, it has a special molecule, and once it polymerizes, it gives off light. And so there's a camera عطول عم تطلع عليهم, the microscopic camera أكيد, and it's looking for the changes in color. كل ما يعلى, one of those molecules is going to give off light. And it's going to keep doing it and over and over and over, and it can do it very, very fast, right? The camera is unbelievably fast in detecting the light, and it can tell والله هلا ضوت أخضر معناتها it's G, هلا ضوت أصفر معناتها it's C, so on and so forth. And it doesn't look at one probe only. It looks at the entire slide. Okay? It does this. Now, eventually, you get enough samples and you get enough of the DNA sequences يلي بنسميهم contigs, and you line them up. How do we line them up? It's very easy. ما الادابترز نحن بنعرف بدايتهم ونهايتهم. So all we do is we take those adapters بنقصفهم, and whatever is between the adapters is the actual DNA sequence, and we use something known as graph theory. Graph theory allows me to sort of like visit each sequence, and eventually you get the entire DNA sequence by aligning them. بنفتش على common sequences وبنلزقهم كأنه عم نلزق a puzzle. Eventually, when you have enough patients, you can create a database يلي فيه بقلبك polymorphisms of a certain gene. Now, next generation sequencing بتزابر. ليه؟ لأنه it uses the Human Genome Project as a reference تتعرف وين بدها تحط بالضبط each sequence. بس مرات كتير you get a certain gene that is mutated, but you have no idea if this gene causes just 1 second قبل ما أكفي. There's a question by Zainab. So we stick to all the fragments obtained from DNA sequencing. Can you rewrite the question وبرجع بجاوبك عليه لأنه I didn't quite understand the question. So تكفي الفكرة. So sometimes you get a DNA sequence مش عارفين إنه هل هي اللي سببت الديزيز اللي عنا أو لا. خصوصا إذا كانت a SNP, a single nucleotide polymorphism, and you don't know if these polymorphisms are linked to disease or not. But once you get enough patients, you sort of can can get an idea إنه إذا إيه أو لا. المشكلة بهول databases إنه they are proprietary. They are like each lab has their own database, and the databases are extremely expensive. صورة الداتابيس اللي عم فرجيكم عليها contains around 100,000 records, something like no أكتر شوي. It contains تقريبا if I remember correctly شي 120,000 record, and this database can sell for around $500,000 because this is accumulated knowledge over years, and it's very expensive. Now there are public databases. You can go to NCBI microarray gene expression databases. These are free. They show you all the measurement data, but they are not as accurate as the databases يلي the good labs بيكونوا مطلعينها هني. Okay. So here's the thing. If we know your genes, we can now know what medication to give you. Okay, give you an example. Depression في around 5 generations of SSRIs. It's a medication نستعملها for depression. غير mood stabilizers. So there's a lot of medications. Which medication is best for you? We have no idea. بدنا نجربهم كلهم. بينما إذا عملت أنا DNA sequencing, I can tell you exactly which medication you can take. The example اللي عطيته بالأول يلي هو الميثيلين